My name is Daniel Feeny. I am a student at CU Boulder, and I am at the PHAGES 11th National Symposium. For my and my lab partner's research, we looked at cluster O Mycobacteriophage Blessica, and one of the main reasons that we decided to look at this one, which our class annotated, was because, first of all, they're very unique in that they have this long prolate head and an especially long tail, which is very unique for phages, but also because they can be difficult to study because all 15 of the O cluster phage have about 97% nucleotide identity, so they're so similar that they cannot be stratified into subclusters. So comparing them to each other and trying to find, meaningful ways to look at them other than just these data, like these numbers that say they all look the same, is pretty important. And as we were looking at them, one thing I noticed that they either have regions of complete similarity, as you see here in the purple, or regions of no similarity, rather than partial similarity or some nucleotide identity. And when you look at these regions of no similarity, as you can see up here, they are either insertions or deletions of these groups of 1 to 3 genes. And that got me thinking about horizontal versus vertical gene transfer. So horizontal gene transfer is when it's not a direct result of parent to offspring, but it's actually the result of gene transfer from one organism to another without reproduction. So that's what I ended up looking at, because I thought that even though statistically they look very similar, this could be a useful way of examining or comparing these phages to each other, and even outside the cluster. So I started with trying to find all of these locations of insertion and deletion as compared to Blessica as kind of the base for finding these differences. And I found things like here in genes 77 and 78, which belong to Dylan, a different O cluster phage. They are not in Blessica, but they are in 5 of the other 8 O cluster phage that I've looked at in our research, and they are only conserved in the O cluster, as opposed to some of these other ones like gene 82, which while it is in Blessica and about 4 other of the O cluster phage that we looked at, it is also in every single L1 through L4 cluster phage. So the odds that almost every other, I think 62 out of about 65. But what that means is that it's probably much more likely that it was a result of horizontal gene transfer from outside the genome and a more recent evolutionary event. So that kind of gave me the idea of expanding upon this and finding all of these locations of insertion and deletion, and being able to identify, what is the most native O cluster phage going to look like? What is a potential common ancestor? And that is this figure up here at the top. So it is my construction of a hypothetical common ancestor. The green boxes that are spread throughout the femurator image at the top are regions where I had to insert highly conserved families of genes into Blessica's genome that I believed are evolutionarily native to the O cluster. And the red lines are like gene 82, which are prevalent in many other clusters, or especially one cluster, and not that well conserved in the O cluster. So they were probably a result of horizontal gene transfer from outside and more recent, so not part of that native hypothetical common ancestor that I'm trying to construct. And I used that and just general comparisons of the locations and numbers of insertions and deletions between the O cluster to create these 2 figures down here. The first one is a relative phylogenetic tree as compared to Blessica, our O cluster phage. So looking at the various numbers of differences and basically these maps of insertion and deletion. And Jane Dynasty, I believe, had only 3, while Dylan Zaki had 13 regions of difference compared to Blessica. So I created what I thought was a pretty useful way to create relationships between the O cluster phage in ways that a lot of the other bioinformatics tools were doing differently. And then I also did a similar comparison of these O cluster phage to the common ancestor I created to try and find the oldest and most ancestral O cluster phage. Again, it's relative. The distances aren't exact to their relatedness, but I created with the O cluster phage, we looked at a tree showing the ones that had the most of these recent evolutionary or like genetic gene transfers. And I thought that this was pretty cool, but I wasn't exactly sure that my method of comparison was accurate. So I looked at a few different factors. I looked to support, the evidence of this truth. I looked at promoters, promoter variation in the O cluster, because they come in one of two variations. They're highly conserved throughout the whole thing, except for in the start here. They either have one forward-facing promoter in this location or two forward-facing promoters. And I assume that if they have similar promoter variations, they're more likely to be closely related. And this was generally true. With, as you can see here in this relatedness, it'll also come up down here. The cat dog, Jane Dynasty, and Blessica, which are all I found to be closely related, also have the single promoter variation. And then young Jamal, corn dog, Dylan, and Zaki, which are up here at the top, have the double promoter, which suggests that my comparisons of relatedness is pretty accurate. Again, also these break-off lines mean that they were especially similar with very few differences of insertion or deletion. And I thought that this was pretty strong supporting evidence, and it also lines up down here. I won't go through it, but I wanted to know, for sure, maybe because it's, recent horizontal gene transfer potentially, maybe location plays a role into this, and it can also support the relatedness trees I created. So I made a map of where each of these phages were isolated from, and again, this information pretty closely supported the relatedness relationships I came up with. So here in Pennsylvania, all of these orange dots are very actually closely related phage with very few differences of insertion and deletion, and generally in similar locations as compared to Blessica. So that was pretty strong supporting evidence, as well as Dylan and Zaki being extremely similar. So they probably, it's potential that they evolve closely together. So again, that's another piece of supporting evidence that I was able to find. And with all that, I actually think that it's pretty valid that I was able to draw comparisons in a slightly different way, looking at these insertions versus deletions. And I think that it can be a helpful way for, specifically the O cluster, where this is pretty unique, and it's much more simple than other phages, which can become more complex when you're looking at different aggregator maps, obviously. But from what I can tell, I did find some pretty strong supporting evidence that this is a useful way to compare a cluster phage.